Okay, time for a couple of videos about the immune response. And when it comes to immune response, a couple of big differences. Remember that if we're talking about a bacterial cell versus a viral infection, we're more likely to find the bacterial infections outside of the cells, so in interstitial space, in blood, places like that, whereas the viral infections have likely gotten into a normal cell, taken over the nucleus, and are producing copies of the new virus with inside that cell. Okay, so if you think about the immune response in terms of steps, regardless of whether we're talking about the humoral response or whether we're talking about the cell-mediated response, the first step is always going to be the same. Step one is always going to be some sort of recognition. Okay, we have to be able to recognize that the infection is inside of our body, right? And so we always do that by looking for, with that recognition phase, we're always looking for the antigen as a marker of something that's not our own cell. So see, it's a three-dimensional shape. Our own cells have our own antigens, but what we would be looking for would be a foreign non-self antigen, right? So I've kind of simplified these into three-dimensional shapes. So the bacteria has a triangle-shaped antigen, the virus produces a circular antigen, and our own cells have kind of a square antigen, right? Now, recognition is going to be different in the humoral or in the antibody-mediated response. What you would have is, in general, you have some sort of B cell which releases free antibodies, and free meaning they're free in the bloodstream, and the free antibody would do that recognition. Whereas in the cell-mediated response, what we would be looking for is either the T helper cell or the cytotoxic T cell to be able to recognize that foreign antigen. Okay. Step number two, once recognition takes place, that triggers cell division. Okay, and as a result of cell division, we create two sorts of cells. We create the effector cells, and in the case of the B cells, you're going to create effector cells which produce more antibodies. So the more effector cells we have, the more antibody is produced over and over again. The last step in the cell division phase is that we create memory cells, and the memory cells are important because the memory cells, they don't produce an active response, but they produce a memory, right? That's why they call them memory cells. They produce a response the next time. So the next time we see this antigen from the bacteria or this antigen from the virus, it stimulates a more rapid response because we have these memory cells to be able to create more effectors later on. And then the last step is the actual response. Okay. Now, in the humoral response, in the humoral immune response, antibody-mediated response, when the antibody binds, that triggers complement, and complement starts poking holes inside this bacterial cell until bacterial cell breaks apart. On the other hand, in the cell-mediated response, depends on whether it's the T helper cell or whether it's the cytotoxic T cell. So the cytotoxic T cell is the most active cell, and when it finds this cell, it's just going to launch a salvo and destroy this one infected cell. The role of the T helper cell is it will activate cytotoxic T cells. The benefit of the T helper cell is that the T helper cell also contacts B cells. And so at the same time that we trigger this response, where we're destroying the active cell, we're triggering cells that produce antibodies. These free antibodies can help now trap the viral particles as they're trying to leave that cell as it's breaking apart, as it's going through lysis. So the T helper cell ends up being the most important cell because it links the two responses together.